So the whole grant process has an awful lot of paperwork associated with it. Um, that every single step seems to have hurdles, even once you've already gotten a good grant score and they have put you on the list to be funded. Welcome everybody. Let me just see something real quick. Look on the. So I just want to see who's on. Okay. Okay. So welcome everybody. So today that I am focusing on how to write kind of when people think of grant writing, what they think about is the topic of today's lecture, which is the whole idea of doing your specific aims page and developing that strong research plan. So, this is mostly an introduction to how you should think about all of the different descriptions of your research that you're planning, that you're going to be submitting to the NIH, and how to write them and how to think about writing them. Now, the big thing to understand is that in any typical NIH research grant, you're going to be describing your research in 4 different places. And in each of those different places, you're going to need to remember what the audience is and remember the tone that you're going to want to take with those audiences. And very often, especially for new grant writers, there's a real misunderstanding about what these different pieces are for and who reads them. And that misunderstanding can make people put effort in the wrong locations and even sometimes put the wrong information or use the wrong sort of language in the different places. So I want to go through these 4 main areas, sort of the way to think about them. And then at the end, I'm going to go through a specific aims page of a grant of mine that actually just recently hopefully will get funded if I can get through all of the NIH paperwork. But it's on the actual funded list. So you can see at least my philosophy for writing specific aims pages. One thing that you should understand is that there are many different approaches you can take to write all these sections that have been successful for people. That I'm going to give you my opinion on how I approach the problem. Other people will give you different opinions. But in the end, what you really need to do is ensure that you are an effective communicator. And that's really the whole key to this process. So the 4 places that you're going to be describing your research are the project summary, the public health relevance, the specific aims, and the research plan. And your project should stand alone. Like in each of these sections, somebody should be able to read just that section and have an understanding of what the research is. The first one is that I'm going to talk about as a project summary. Yeah. Yep.
It's Erin. Sorry, I don't mean to interrupt, but we're not seeing your slides.
You're not seeing my slides. Okay, so let me try this again. All right. Share screen. All right. Oh, I see. It's sharing. Start sharing. Okay. You are sharing entire screen.
Now.
So
we're good now.
Do you see slides? Okay, I just pushed the wrong button. This is the new version of, I just went to the new version of Blue Jeans. Okay, cool. All right, so all you guys missed was that slide and just the description of the project summary, the public health relevance, specific aims, and research plan. So the project summary is that sometimes a new grant writer will think of the project summary as an abstract, like if you're going to the abstract of a conference. However, the audience of the project summary is not the same as the abstract as the rest of the grant. And the project summary's audience is basically the public record. And you should realize that the project summary is going to be posted on the NIH website. It's going to be accessible to the general public. And while the readers can be the research community, and as in the last lecture when I said how to communicate with the NIH, I suggested you went to NIH Reporter and look for who's funding your research. The project summary are the sorts of things that you see in NIH Reporter so that you're seeing other people's project summaries. It's also the general public. Very often it's the press. NIH, sometimes it's NIH administrative staff. A very important constituency that reads the project summaries are Congress and congressional aides, members of the study section who review your resubmission or your competing renewal, because the project summary is the only piece that gets included in your reviews. So that's the only piece that a study section member will see on a resubmission or a competing renewal. Because this is what's included on the summary statement of when you get your scores back as part of the scope of work. And the format of this is a standard unstructured abstract where you just lay out what you're doing. However, because its audience is the general public, you're not using the project summary here to actually describe the nitty-gritty details of your research, or even necessarily to be all that specific about your research. That you're going to want to use most of the space to describe the problem and emphasize why it's important. And it's because these are the public project summaries that tell the world what NIH is funding. You want to be very, very careful to be aware, since it is in the public record, that you want to avoid anything that looks politically controversial or could look bad to the lay public. So that this is why you want to focus on why it is globally big picture important, because this is the only public face of the grant. This is when you listen to Congress talk about silly waste and federal grant funding. These sorts of summaries are where they get that information. Where there was a very famous case where this was used is something called the Proxmire Golden Fleece Awards. So William Proxmire was a senator who back in the 1980s had congressional staffers read project summaries for funded NIH grants, and basically came to the conclusion that, oh, these projects are silly. Why are we wasting money studying certain things? When it was just that the description of like the research was very specific and didn't actually emphasize why anybody would care. And this allowed some really important research studies to get targeted as silly waste, fraud, and abuse. So you want to make sure that these project summaries make it extraordinarily clear why your work is important, because when your grant's funded, nobody wants to have to deal with reporters and having to deal with, getting interviewed because a congressman has targeted your research as something being stupid. And it's because, very often you read the description of what you're actually doing. Like I study cataracts in mice. Well, who would care about mouse cataracts? Who wants to do that? Well, it's because mice are an animal model of cataract disease. So you have to make it really, really clear the importance of what you're doing. Don't just lay out, the details of what you're doing. Also, you want to consider that it's often best to keep the description of the proposed work rather vague. Very often you don't want to say, I'm going to do X in specific aim 1 or B in specific aim 2. Some of this is that since it's the public face of the grant, is that you don't want to tell other people necessarily in great detail what you're working on. This is a place where, somebody could try to steal your idea and maybe scoop you on the project. It also, if you're too overly specific in the specific aims, it could make it more difficult to get your award renewed. And this is because the reviewers of your competing award renewal only have the project summary as their only guide, and the prior reviews as their only guide of what the prior award was focused on. And while we all know it's normal for the scope of work to change over the course of a 4 to 5 year grant, you want to be as flexible as possible on what to include in that scope of work. So because you gain new information, you're going to change your scope of work. But if you're overly specific, then you might find yourself needing to explain why your scope of work changed when it really didn't. It was just that what was important changed over the course of your grant. Now, and in general, the project summary is, it's something you write in the couple weeks before the grant's due. It's generally not something that you need to over-focus on. Once you have your grant actually settled and what you're going to be writing your grant on, you write the project summary as just a very straightforward discussion of what you're doing and why, and emphasize the why. Now, the next piece of the grant that is also publicly available is the public health relevance section. And again, this is the other piece available to the general public, and it's posted publicly on NIH Reporter. Now, the readers of this, again, is the research community, the general public, the press, NIH administrative staff, and again, Congress with 3 exclamation points. This requirement was added to NIH several years ago to, again, avoid the Proxmire effect, that they want some, especially people who are working on interesting models that might not be immediately obvious have public health relevance. Like if you're working on sea urchins, or you're working on plants, if you're working on C. elegans, or the small nematode animal models, that the actual research description may not explain what the reason why NIH would fund research on a plant. Why is NIH funding on a plant? Well, it's because it's a really, really important model of microRNAs. But that might not be obvious from the project summary. So the public health relevance is asking you to explain why your project essentially is not waste, fraud, and abuse, and why is NIH funding it. So this is to describe the relevance of the research to public health. It is most 3 sentences. So you can either describe how, in the short or long term, the research contributes to fundamental knowledge about the nature and behavior of living systems. That is part of NIH's scope, is basic biology. It also, though, is how your project could enhance health, lengthen life, or reduce illness or disability. That this is a part of proactively educating people in relationship to the importance of basic science research, but it also can be important if you have a study that seems to be common sense, if you're doing like a sociological or a psychological study, where it's like, well, everybody knows people act in a particular way. Why are you spending my hard-earned tax dollars on that? Well, you use the public health relevance to explain why we actually have to use research to research topics that other people might say, oh, it's common sense, it works a certain way. So that I'm going to leave those aside. Both of those are usually written in the week or 2 before the grant's due. That yes, you need to do them well. You want to have them being clearly written. They should be written so they're definitely intelligible to people who are not experts in your field. Now, where the piece that could arguably be the most important section of the grant is the specific aims page. And the specific aims page is a one-page, single-space document that is really, really important to your grant. The thing that I can't emphasize enough is that the specific aims page needs to stand alone. And it needs to be completely intelligible, clear, and self-contained, because many or most of the people who are going to be making decisions about the funding of your grant will only be reading the specific aims page. They will not be reading the rest of the grant. The audience of the specific aims page is mostly internal to the NIH, but these include the people who make the decision about what study section your grant will be reviewed by, which can absolutely make or break a grant. The scientific review officer, because the scientific review officer reads that specific aims page to make decisions about which reviewers will be assigned to your application. Program officers. Program officers are reading the specific aims page to figure out what portfolio of funding that your grant is going to fit into. And at the institute level, which even program officer is going to be administering your grant, and then the program officer, once it's assigned to them, is going to use it to decide what funding portfolio and which budget line they're going to be putting your grant towards. Study section members. Remember when I talked about, finding a study section, that the thing to realize is that 3 members of that study section are going to be reading your grant in its entirety. However, all members of your study section are voting on this, and those non-reader study section members will essentially only be reading your specific aims page. And their vote is equally powerful in the final impact score as that of the study section members who are reading the entire application. Other NIH administrative staff, as well as the Board of Scientific Counselors. So I don't know if I talked about who these people are, but the Board of Scientific Counselors is it's considered the second tier of NIH peer review. So grants are initially reviewed by the study section, but all grants are then secondarily considered by the Board of Scientific Counselors, which are a group of recognized senior subject matter experts in your area. And the Board of Scientific Counselors, in some ways, you could consider that as they're almost rubber stamping the decisions of the program officers and the study section, but they also have the ability to say, oh, no, we shouldn't be spending money on that, or, oh, this grant, which was, in the gray area or just outside the pay line, is such an important area, we really have to make sure this is funded. And the Board of Scientific Counselors are basically reading the specific aims pages of all the grants that are coming into the Institute. So you want to have these be very, very well written and clear, and make it so it's dead certain what the importance of the work is. Now, the format of these are one page, and you can think of this as a one-page technical abstract that has to stand alone. It has to set up the scientific problem. It has to address what questions you plan to answer, your hypothesis, and your set of objectives. And these set of objectives are your specific aims. And so from reading this, that they should have such a clear understanding of what this grant is about. Somebody can make the decision of, do I have the technical expertise to review this or not? If you're a science study section member, you're asked, are you comfortable reviewing this? People make those decisions based on reading the specific aims page. That basically this has to be complete technical judgment. Whoops, I didn't mean to do that. Okay. Now, some tips to think about writing that. So it has to be a standalone, freestanding document that is understandable, even if you don't have access to the grant body. Now, how I structure these is the idea that the first paragraph clearly describes the scientific problem and its significance so the reader can immediately understand the who cares. Now, this should be understandable for somebody who might not be a direct subject matter expert, but it's somebody who is probably master's or doctorally educated in the life sciences in some sense. But it shouldn't, so you don't have to worry about it being readable by a high school student, but you do need it to be generally understandable for somebody with a general technical education. It then needs to include the specific aims proper, which outline 2 to 4 objectives that the research accomplishes. And please, please, please use the H word. Specifically state a hypothesis on the aims page. And it's even preferable to state them several times. It can be very powerful to state the objectives or your aims as hypotheses to be tested. Preferably, you can have this supported by some clear evidence supporting that the idea is true. And it can also be very powerful to state each specific aim as a hypothesis to be solved. Now, some mistakes that I see people make is that they do in-depth technical descriptions of how they're going to accomplish the aims. This is not a place where the reader wants to hear about the Western blots they're doing. They want to understand the objectives and how those objectives will be accomplished to move the field forward. That one of the articles that I gave Erin to post on the site is that I particularly love. It's called Your Grant, the Rejected Recipe or the Funded Menu. And it sets up the thought experiment of if you're a hungry restaurant patron and you walk in the door and the waiter can either hand you a stack of recipes that start with raw chicken, or they can give you a menu that describes this lovely dish and how it's going to taste and how it's going to feel. The specific aims page should read much more like a restaurant menu than the stack of recipes that you are going to have to envision how those recipes are going to come together to a final product. In almost all cases, make sure that the aims of the grant actually test the hypothesis. So if you have a global hypothesis and your actual experiments and the aims of your grant don't actually address your global hypothesis, that's a really, really big red flag that'll get your grant not scored well. And this might seem obvious, but it is an incredibly easy trap to get into. That you write a very compelling hypothesis, but then you start writing your experiments on things that you know you can test. But in the end, once you've written your grant, that the experiments that you've proposed don't actually test the global hypothesis. And that's a place where you need to really think about either reworking the experiments or reworking the hypothesis to make sure that your experiments are directly testing it. I have gotten myself in trouble with grant applications so many times because of this thing, where I have set up a very compelling hypothesis and then my experimental plan in the end never really got at the crux of that problem. And that can really, even if the experiments are great, that can really kill a grant. And also, because of the way NIH is now scored, NIH is based on these impact factors. And the reviewers of the grant are asked to judge really what's the potential impact of this. If you did all this work, what is the potential impact on the field? That this is something you should be able to very, very clearly articulate in 1 to 2 sentences. So it's this very short takeaway explaining how the proposed work will move the field forward. And don't be afraid to be explicit and call it impact, because impact is the score driver of an NIH grant. Now, I'm going to get out of the main presentation and open up, see if I get into the right document that I wanted. And I should have had this prepared. Hold on a second. Okay. All right. So I'm going to go to an example specific aims page. I did upload this. I'll give this to Erin. This is the specific aims page of a grant that I submitted last summer that received a fundable score, and hopefully I will get an award letter for this week if I can get through the just-in-time process. So I'm just going to walk through this kind of from a philosophical point of view. So what I started with was a big picture question that basically talks about how the lens works. So this is just, I study cataracts and I study the ocular lens. So the first couple sentences just talk about how the lens works is we have cells that communicate with the basement membrane, and that the communication of how the cells talk to this basement membrane affect how lens cells behave. And then I talk about how this behavior gets messed up in a condition that arises after somebody undergoes cataract surgery, which is called posterior capsular pacification. So then I set out the overall objective, and my overall objective is determine the mechanisms by which how cells communicate with their matrix influence the pathogenesis of the disease. Now, that then set up my following specific aims. Now, this grant sets up, my first aim is stated like a hypothesis. So it tests the hypothesis that this protein is important to regulate this medical condition. And then I set up under this aim the premise. I basically talk about why I think this molecule could affect this disease. So I'm then saying is that I'm testing this particular hypothesis and why I'm testing it. In my second aim, this is more related directly to a mechanism. So I basically say that I have made this discovery in the past, and I'm understanding the mechanism that this works. So I'm setting up the problem as how this is the function of this molecule, that I said that this is what we've discovered, that we have also found that it's involved in this particular cell signaling process, and that I'm here directly investigating the mechanisms. Now, looking at this would have been better if I have stated it as talking about a hypothesis. So instead of here we investigate the functions, I probably should have said something in relationship to we test the hypothesis that the direct interplay between fibronectin and this molecule are important. That would had been a much better way to do this. But this is a case where I know what best practices are, and in the heat of the moment writing this grant, I did not state this specific aim in retrospect the way I maybe should have. So this is a case where it's really easy to get into your own cognitive traps. Now, later on in the actual grant, I do specifically state hypotheses, but ideally I should have stated it on the aims page. But the important bit here is impact. So here I'm stating flat out why this is something that's important. So I am basically saying that this work in this one fibrotic disease could be important in many, many fibrotic diseases. So I'm talking about the impact of this work is not just beyond my little fibrotic disease. It could be important for all sorts of fibrotic diseases. And I say, why is fibrotic disease important? That I'm talking about why that we need new studies involved in fibrosis. I'm saying that why my preliminary data is related to one model of fibrosis, and how if I can figure out how to block these pathways in this model of fibrosis, it could be important in all sorts of conditions. So I've basically really tried to tie in not just a molecular detail of how what we're studying is involved in this little teeny scientific problem. I'm trying to relate how what I've learned is going to make a larger impact on the field or even in conditions beyond the one I'm studying. Okay, so that's sort of an idea of how at least I approach specific aims pages. It's probably good, though, to directly talk to your various mentors and colleagues who you know have been successful in the NIH grant process, because there are definitely many, many styles and forms and flavors that you're going to see with specific aims pages. Okay, so do I have any questions about specific aims or the other sections that I've talked about so far? Can I go on?
Hi, this is Evan. Can you hear me okay? Hey.
Yep, I can hear you fine.
Perfect. I had two questions, and I'm sorry if everyone else knows the answer to these. Do the project summary or the public health relevance contribute in any way to your overall impact score? Or are they irrelevant for how your score is, how your grant is scored?
Okay, so this is a case that depends on the individual reviewers.
Okay.
To be perfectly honest, that I've reviewed for NIH for 15 years. Over that time period, I would say that I almost never read those sections of the grant. Okay. That I might have glanced at them, but I certainly never read it in detail, and I never based my opinion of a grant on them.
Okay.
That said, that the reviewers do have access to those pieces of the grant. So I'm not willing to tell you that they're irrelevant, because individual reviewers are going to put different emphasis on different things in the as they read, right?
Yeah.
Personally, though, I never bothered.
Okay.
But so I'm not. You shouldn't certain plan on doing a crappy job.
Yeah.
That the place where that research description is read in detail by reviewers actually probably isn't in the review of this application. Where it is that if you receive R01 funding
Mm.
and you're going up for competing, either let's say you have it reviewed and you go in as with a revised application. The reviewers of the revised application will read the project summary from the prior submission,
Okay.
because that'll give them an idea potentially of what the scope it was of the prior submission. Because they'll have the reviews of the prior submission and they'll have the project summary of the prior submission, but they will not have the body of the prior submission. The other place is that if you get your R01 and it goes up for competing renewal in 3, 4, 5 years, however much money they gave you, that the project summary will be the only that the project summary and the reviews of the funded award are what the people reviewing the competing renewal will have in front of them when they review the competing renewal. So that's another place where that project summary is being done. From the standpoint of the public health relevance, the people reviewing it are probably very, very deep subject matter experts in your field. So they won't be reading the public health relevance simply because they won't. They already know. They're either convinced or not convinced. Your public health relevance isn't going to change that. Does that kind of make sense?
Yeah. And this is totally not the point of the discussion today, I know, but just at the 10,000 foot level, what determines whether your renewal is a competing or non-competing renewal?
Okay, so a competing renewal. So when you get a grant award, that they will if you're submitting an R01, for instance, you should, especially if you're new investigators, you should ask for 5 years. Period. Most new investigators will be awarded 5 years. That somebody like me, who's senior, I almost never get 5 years. They usually cut me to 4 and occasionally 3. Because NIH's policy is the average grant has to be 4, so for every 5 they have to give it 3. So like for instance in this grant I'm hoping to get my award letter for, the program officer told me on the phone they're going to give me 4 years of the 5 I requested. So that this time next year I'm going to have to send in a progress report. And as long as my progress report is submitted and everything's fine, then they're going to give me a non-competing renewal. So it doesn't go through the review process. It doesn't go through study section. It's just the program officer is going to say, yeah, she's doing the work she said she'd do. All looks good. She's complying with everything she should be complying for. We'll give her the award. That's the non-competing. That, however, in 3 years, 3 months, I then need to apply for competing renewal. So they have only promised me 4 years if I'm doing a good job. In 3 years, 3 months, that if I want to keep my grant current without a break in funding, that's when I'm going to be submitting a whole new R01 application that then goes in and is reviewed by study section. So that's a competing continuation or competing renewal. Does that make sense?
Okay. Yeah.
Okay, cool. Right, any other questions?
Melinda, this is Andrew Matuskowitz. I do have a question, if that's okay.
Hi. Absolutely.
Thank you. Very specifically about the hypotheses. I was reading a web page from the NIH about the specific aims page and how to construct it. And one of the pieces of advice, and I shared that with Erin, by the way, and I thought it was extremely helpful. And so I encourage everyone to take a look at it as well. But one of the things that I noticed that was I found very interesting was it's suggested that when describing your hypothesis, it recommended against stating exactly what you expect the outcome to be, and it recommends instead focusing on how you expect your research to help you better understand the relationship between your between different things.
Okay.
And so I just found that
Okay,
really
so
interesting and was curious. Do you find
sorry,
it a bad idea?
sorry a minute. I'm just trying to shut up my cell phone. Very
Oh, no
button at
problem.
first. Sorry.
So the reason that they're suggesting that is that if you make a very specific hypothesis without information, that you can run into the trap that the reviewers may feel that you have such a strong view of what the answer is going to be, that you have such a strong review thing of what the answer is going to be, that you're going to have a preconceived notion and you're not going to be doing research where you have an open mind about what the outcome is going to be. So that's
Right.
definitely a risk of your hypothesis being too specific. The problem is that if you are, doing a hypothesis that is too kind of vague like that, I mean, it's not really a hypothesis. You're just saying, I'm going to be studying this interesting thing. So you have to be careful about that as well. So what I would say is, so if you are making a specific hypothesis, then in your research plan, it is really, really important when you talk to really do your alternate outcomes and alternate approaches very, very carefully, so that you acknowledge that if I do this experiment, it may come out in a different way, and if it does, it means that this part of my hypothesis is different. In my specific games page, the hypotheses that I'm stating are actually stuff that I've already proven and I have data for. That it's the mechanism thing that is basically stuff that I can't, I will worry about and can't deal with. Where this is, like I have like these small sub-hypotheses later on in my research plan. I guess I'm kind of talking in circles, but that's the tricky bit. It's definitely very, very risky not to use the hypothesis word, because if somebody says your proposal is not hypothesis driven, even though NIH Central says that all proposals don't have to be hypothesis driven, there's this risk that your reviewers aren't going to buy that, and they'll look at that as a negative. So I realize I'm being kind of vague here, but
no, that's okay, and that's helpful. I mean, the challenge is it does help. So don't make a specific hypothesis that if it ends up being wrong, it ends up not being, let's say it's the null hypothesis, and you're proposing there to be a change between two things. If it ends up being the null, just make sure that you have a mechanism by which you can then continue the studies and not have that be a catastrophe.
Other question, I mean, go ahead.
Yep. The other question is then, do you think, how important is it to use these words, my hypothesis is, do they want you to show them exactly where the hypothesis is, or is a good specific aims page, is that implied by saying that, we hope to demonstrate these two things?
Well, I mean, so I guess how I would look at it is that you set up a global objective. And the global objective is you're trying to answer some kind of critical question, whatever that could be. Now, when you form a hypothesis, you basically are and the idea is that you're basically trying to test whether your hypothesis is true or false. And that you have to make sure that your experiments will get at both not just confirmation of it being true, but could give you that your hypothesis, you actually will reach the null hypothesis. So you kind of have to make that clear. That, I guess how I kind of like the structure is that a specific aim is to test a particular hypothesis. I mean, and you can't just pull hypotheses out of thin air. You have to have hypotheses be, have significant amounts of support, because then that's where, the reviewers will say, oh, you don't have preliminary data for that. Like you're just guessing. So, you usually have hypotheses that have significant experimental support associated with them. They're not just, something that you pulled out of the literature or something like that. Does that kind of make sense? Or is that, am I getting at what you're asking?
No, that is helpful. And I didn't realize that it's important to try to have background data from your specific study, from your specific institution in your specific aims.
Well, it can be helpful. I mean, it's a style thing. I have definitely seen specific aims not use that style. But if you can if you have a really key piece of data that is saying that this is why this is so exciting, it's not that you pulled this hypothesis out of thin air, but, that you think that you have this important problem that you now have a hypothesis that it's working this way, and you have a key piece of supporting information to suggest that, yeah, this really is working this way. Please give me the money to really, really nail this down and flesh this out. I mean, that's sort of the ideal. This is the hard part, actually, especially for new investigators, is because and new investigators do get a break, that you're going to be allowed to go in with less supporting information than somebody like I would be. That said, you also, want to make it really, really clear that you're not just making stuff up. You actually have in your data generated by your own hands to suggest what you're proposing might be real.
Gotcha. Thank you. Thanks.
And
that helps.
If that can be conveyed to the study section, beyond the people doing the review, that's why I personally like to put one or two pieces of key data justifying the aim. But that's my stylistic preference. You will definitely see funded grants with aims that don't look like that. And you're going to kind of have to, it really helps a lot to get your hands on as many aims pages as possible, particularly from funded grants, because you're going to see a whole range of styles. This is actually why getting asked to be an NIH reviewer can be so powerful, because then you'll have access to 100 grants, and you'll be able to see how different grants get treated and read by study section. And that's a really, really helpful thing to be able to experience when you're trying to figure this out. But if you don't have access to being on a study section, by just talking to people and seeing if you can get their aims pages is really, really helpful. Is that good?
Very much. Sorry, everyone, for hijacking Melinda's time.
Oh, no. I think I think I actually prefer to ask questions here. If there's anybody else with questions, that'd be cool. Because keep in mind that.
Dr.
Go
Duncan,
ahead.
it's Evan again. I was going to jump in and say, I think as far as the preliminary data question goes, there's there's a section of the grant that is specifically for preliminary data.
No, there is not. Not in an NIH grant. There is not. In a preliminary data has to be that section was removed. The preliminary data gets woven through the research plan. There is not a preliminary data section in an NIH grant under the current rules.
Oh, okay. Did there used to be?
There used to be, when I first started grant writing, there was back 20 years ago, but this got removed when there was the redoing peer review, maybe about 10 years ago, 15, something like that. I think it was probably about 10 years ago, maybe a little bit more recently than that. But I'll
Okay.
explain, it might be what you're calling preliminary data, but let me show you from the research plan point of view. Let me go through the different pieces, and I'll talk about how personally I handle preliminary data, but there is not a section of an NIH grant called preliminary data, even though there used to be, back in the 90s into the 2000s. But that basically went away as soon as NIH went. In the old days, research grants were 25 pages. Now they're 13. And when that change was made, the preliminary data section got removed from NIH grant. Kind of talked my way through this. And if you still have questions about this, let me know. That sound good? Okay. So for the research plan, the audience of that to be perfectly frank, the only person who's going to read that in any depth are going to be the 3 reviewers assigned to your grant. It might be skimmed by others on the study section if they want clarification of something that they saw on the specific aims page. But in most cases, you're going to be putting your heart, love, and soul for at least a month, probably longer, into a document going to be read by 3 people. Now, this research plan, it's 12 pages long, and it's separated into 3 sections. The first is the significance, the second is the innovation, and the third is the approach. So note, there is not a section that talks about specific aims. That's not specific aims. I'm talking about preliminary data per se. However, you need to give preliminary data woven into these sections. Now, this is the meat of the proposal. This is what's largely going to drive the written reviews of the grant application. It's going to drive the biggest opinion, and by far the research plan is the biggest score driver, because this is what's going to be what is used to actually write the summary statement and to drive the scores of the people who are reviewing your grant. Okay, so the first section is the significance section. So this is the opportunity to elaborate on the big picture scientific question. It's the introduction to the field. Generally, the tone of this is very important. You have to be aware of who's reading it. That I had mentioned about trying to figure out what study section might see your grant. This is a place where it can be extremely helpful to look up the background of those permanent study section members of your grant. And you want to be able to write the significance and put themselves into their shoes and present information that reviewer pool will buy. You want to think about what are their preconceived notions in the field. You want to think about what things is this group of people likely to find interesting and important. That it is essentially saying it basically is the long form of setting up the big picture scientific problem you're trying to get at. Personally, though, and from my grant writing approach, personally, I only use the significance to set up why I am studying the big picture objective. I do not use this as a section to sell the significance of my individual aims. And the reason for this is that a 13-page, 1 page of specific aims, 12-page research plan, single-spaced, is a fairly dense document. Reviewers may not read your grant all in one sitting, and they may not remember stuff you provide in the significance when they're trying to understand the approach let's say your third specific aim. So I recommend that a decent amount of the significance of an individual aim be woven into the approach section as you describe each aim. And that's also where I put the preliminary data for each aim largely. Bury that within the aim under the approach section. I might put 1 or 2 key pieces of preliminary data in the significance section, but I typically don't put a lot of proximal detail there. And because of this, you have 12 pages to tell this whole story. I personally recommend the global significance of the project. Try to keep it about a page. And then it's not that that's all the significance you're going to apply. It's just that the aim specific significance. So for instance, your big picture significance might be that, I'm studying this disease. This is important for these people. We know these things about the disease pathogenesis. However, there's this big hole in our understanding of the disease. And this is your line item verse saying, justifying and proving that there's this big hole or that people aren't considering certain aspects of the disease. And that's where you end it. And the reason you write it is that this is what convinces your reader. Remember, it's the 3 assigned reviewers, which are probably subject matter experts in your field, that you have identified an important knowledge gap in this area, and that this important knowledge gap is something that we really want to fund somebody to fill that knowledge gap. And at the end of the significance section, they should be convinced that you have found an important knowledge gap that really desperately needs to be investigated. Okay, so the next section is the innovation section. Now, the innovation section is actually really important for you to write well and to really be able to put your finger on it. Now, the reason is that innovation is a scorable criteria for NIH grant proposals. Now, NIH has done some studies about the correlation between the various criteria scores and the overall impact score. And the innovation score itself has a relatively small effect on that final impact score. But even small effects are important. However, when that really it's the approach that is the biggest piece that is scorable. But everything counts. And ideally, you want to explain how the application challenges and seeks to shift current research or clinical practice paradigms. In the innovation section, you describe novel theoretical concepts, approaches, methodologies. If you're using cutting-edge instrumentation that no one has applied to the problem, or you're trying an intervention that nobody has described, or advantages over existing methodologies. You also want to explain that you've refined things. You're doing something different than anybody has ever done before. Now, the length of this, it's relatively short. They actually don't explicitly give it in the application instructions, but confine yourself to 4 to 6 sentences, like a third of a page tops. Mine are usually more like a quarter of a page. And this is a case where the reviewers are asked, is this application innovative? The innovation section gives you, the applicant, the opportunity to drive that conversation, to specifically tell the reviewer what is innovative about your proposal. Because if you can't come up with what's innovative, they certainly can't. So definitely write this well, definitely put some attention to it. Just keep in mind that innovation is, as a criteria score itself, is probably not the huge score driver. Now, without a question, the most important section of the grant is the approach section. This is
Can I ask, sorry.
Go ahead. Go for it.
Sorry, I have a question. This is Nira about the innovation, because I struggle with this at times. I think particularly in the context of if there's a study that where the knowledge to be gained may be more incremental in nature, or it's building off of something that's already known, but you're studying it in a different population, or you know what I mean? Because I think oftentimes, at least the type of research I'm doing, the methodology is not going to be innovative. It's secondary data analysis, or it's some type of pilot trial for feasibility. Right. But I think I struggle a bit with how do you authentically talk about innovation when really it's really more about moving, making, incrementally increasing the knowledge to be gained on a particular subject, but it's not something groundbreaking, but it is something that needs to be studied.
Right. So that's always a problem, right? Because since pay lines are so tight with the NIH, everything I'm talking about is NIH specific, right? So there are other funding sources for a lot of different research, but the problem is that there's all that funding is very, very tight. That pay lines, range anywhere between the highest that I know are in the high teens, and the lowest that I know are in the single digits. And everybody applying has the same credentials you do, or more, because they're more senior in the field. So the problem is that very often really, really good science, isn't going to get into that pay line. So this is a difficulty that is something that, sometimes it might seem forced, but it is also a reality of how, reviewers are going to look at this. Essentially that in what I would probably do in your situation is figure out some way to talk about innovation in relationship to impact on the field, right?
Okay.
Because actually, when you review and they call the work incremental, it's almost a poison pill.
Right. Absolutely.
So it's sort of a negative to call a piece of work incremental.
Yeah.
So, so in the end, though, it could be that it is, but if it's incremental, but it will have a huge possible impact, let's say on, I actually forget what your research is, but, I think somebody in this group is talking about whether you put community health clinics in schools. And, it might be that, showing the effective, like the approach might seem incremental, but if somebody can show that that is highly effective, the impact could be huge, right? In the sense of improving the health of underserved populations. So maybe from the standpoint of, it's not what they're asking for, but what's innovative about it is that people have thought for a long time that might be good, but this really will provide the evidence-based data to allow policymakers to make the decision that this should be done. So in that case, what's innovative about it isn't that it's new. What's innovative is that you might be the first person to actually really, really test what seems like is right.
Okay.
So that's just
helpful.
a suggestion. I don't know necessarily if that's going to be effective, but that's a suggestion.
Okay. Thank you.
Okay. Any other questions right now? Okay. So from the approach section, this is the meat of your grant, and it does describe what you're going to do. The format is that you will state your aim. Now, usually when I write these, I start, before I even state my aim, I start with a sort of a recap leading up to my objective. So almost restate the objective and why we are doing this whole project and what is the overarching objective that we're trying to get. Just to remind the reviewer, this is the goal we're trying to meet. Then you state your aim, and that is the objective, the first objective that you're going to try to reach. That you can either state that aim as a hypothesis, or that you will state the hypothesis somewhere here. And then you would give the background, and this is where I would personally recommend putting your preliminary data. So if your specific aim 1 is to investigate the role of something in something, then you could give, that this is an important question that I have, and this is where you can show your preliminary data, whether that is, survey data or gels or westerns or a mouse phenotype or whatever your preliminary data looks like. There you actually give preliminary data as figures. and you say how this preliminary data and this background leads to a particular hypothesis. And then you can explain what experiments that you're going to do to test that hypothesis. Usually, I think that is most effective as sub-aims. So, NIH grants under the current format, they don't want your buffer concentrations. They don't want, detailed protocols. But what they do want is, a description of the experiments that you're going to do. They want a very clear understanding that you know the pitfalls of these experiments, the alternate approaches that you could take if those pitfalls happen. Let's say that, then also you want to describe what the data will look like if the hypothesis is supported or if the hypothesis is rejected. And you also want to explicitly state how you will move forward if that hypothesis is rejected, because they want to be very, very sure that you might have you're doing experiments to see if you can prove a particular hypothesis, but they want you to acknowledge your hypothesis would be wrong, state what the result would look like if your hypothesis was wrong, and then how would you move forward if it was. And this is where it's really important to avoid dependent aims. One thing that's very clear that can kill a grant is that if aim 1 has to be successful before you do aim 2, this is a place that can really kill grants, because if you fail at aim 1 and you have 4 years of money, what are you going to do with the rest of the money if aim 1 fails? So this is a sort of case where you're testing a hypothesis that a molecule is important. You've removed the gene for that, and you're characterizing the animal in aim 1. Well, what if you have removed the gene and there's no phenotype? Well, all of the experiments that are in your next 2 aims don't make any sense because you've already disproven your hypothesis. So it should be that each hypothesis, each aim should be able to somewhat stand on their own. Now, the length of this, the approach section, none is given, but that because I tend to confine my significance and innovation sections to 1.3 pages, that leaves 10.7 pages left in an R01 application to do your approach. And now keep in mind under this format that would mean it's approach and specific and preliminary data. Now, I have definitely other people definitely would sometimes put their preliminary data in the significance section, making a much longer significance section and a much shorter approach section. However, that can be risky because they read the significance section and then you start things in the approach section, and if the reviewer fails to make the connection between what you're proposing in the approach and the preliminary data that was proposed, 8 pages back, I have definitely seen reviews say, oh, you don't have preliminary data to show you can get this to work. But that preliminary data was in the grant. It was just on page 3 when they were reading this on page 10. So that is way less likely to happen if you put the data directly supporting that particular aim, embedding it in that aim. So that's why I personally suggest that. Now, some things that are really important is that you have to explain how those results obtained will have proven to support that hypothesis. You have to be objective and critical. It's also really, really important to keep in mind that correlation does not mean causation. So just because you have shown that, like a place where people get in trouble in my field, is just showing that a molecule is present in a cell or a tissue does not mean that it's functional in that cell or tissue. So you have to be very clear on what is correlation and what is causation, and don't overinterpret what your experiments tell you. And this is a place where, again, make sure that your stated aim, at your experiments actually address the hypothesis and your stated aim. A huge reason that grants aren't funded is that the experiments are not accomplishing the stated goal. Now, another thing that you're going to have to keep in mind, that this was new and added in 2016, you also have to explicitly address how the integrity of the reagents, the animals, cell lines, the antibodies, and also experimental reproducibility is ensured. So this is for social science research blinding, for social science research, really, really good and rigorous statistical plans. Now there is a separate section talking about reproducibility of cell lines in animals and antibodies, but for many of you who do more social science or clinical research, you're going to want to have a very, very clear statistical plan that really, really gets at reproducibility and rigor, because reproducibility and rigor is directly now addressed in grant reviews. So you have to make it really, really clear your experimental design is robust enough that you're not going to fool yourself, or that you're going to be going back and basically looking, basically you have to really show you're not p-hacking. In other words, like your first hypothesis was wrong, so you're going to go back and mine that data 15 billion times looking for something with a 95% confidence interval. That's where social science research doesn't get reproduced. So you have to really, really show statistically how you're going to avoid those traps. It has really become a big emphasis of NIH recently. It also, one thing that you also, once you've written this thing, you should really be able to step back and see how the preliminary data plus the proposed experiments will result in a publication or preferably multiple publications. Because one of the things that they're really going to be looking for is, what is the potential for this, there being an actual product out of that? And that product is going to be publications. And if you haven't proposed everything needed to publish the work, then you're also going to be running into some problems. Also, if your project is in an early stage of development, you want to describe strategies to establish feasibility and management of high-risk aspects. So these are cases where you have to talk about select agents, point out any risks or hazards. But this is also a place where you have to be careful of that you want to make sure that you're, like let's say you've never done a protocol, that you want to make sure you have a collaborator or a consultant who is stating explicitly that they will help you with that approach and that they're expert at it. If you have published on it before, you want to state explicitly in the approach, I have published on this area before, I have done this work successfully before, because that's a high-risk aspect. That, for instance, like I can propose a data mining project on, Medicare data. However, I have zero expertise in that. So even if it's straightforward for an expert, it's not straightforward for me. So that's high risk because I don't necessarily know what I'm doing. But if I have a consultant or a co-PI or a collaborator who knows how to do that, that mitigates that being a high-risk thing. And finally, that each specific aim, I highly recommend you write an impact statement for each aim. And then make sure you end your approach statement on another global impact statement. You can't hammer home the impact of your project too much because, and you particularly do not want to be the last thing that the reviewer reads is a pitfall. You want to make sure that application has a strong finish, emphasizing why that work is critical. Because what you are writing here is that keep in mind that the reviewers are going to read your grant. They're going to do a review of it. They're going to write that review that if they give your grant a high score, that means those 3 reviewers are going to be obligated to publicly present your work and explain it to a group of 30 other people. And they are going to need to clearly articulate why this is the best grant ever and why it's important and why is it going to be paradigm shifting in the field. Your chances of a reviewer being able to get up in public and publicly defend your work is if you have explicitly explained why this is so incredibly important to do as a project. If you can't come up with a very clear, punchy, concise impact statement, it's not realistic for the reviewers of your grant to come up with that for you. So that's something that's really, really important. Now, my final thing I want to end with is that when you're a naive grant writer, 12 pages single space seems like, it's a really long thing. However, in my day, I used to write 25-page grants. That it is way easier to write the old 25-page NIH grant proposals than the current proposals. And particularly, NIH keeps adding new things that have to be addressed. One big one that just got asked, that you have to, if you are studying populations, you have to address sex as a biological variable in your studies. That's a new requirement. They added it onto the existing requirement, and they did not increase the page limit when they did so. So there's a lot of information that has to be packed into a reasonably small space. And so very often you have to decide what you're going to prioritize, what you're going to think about. So everything you say really needs to be thought about explicitly. Now, for all of these reasons, the thing that I would really recommend to you in all of your grant writing, whether it's NIH or any mechanism, is that you write the proposal first draft with no regard to the page limits. Make sure you write and explicitly explain all your ideas. Don't go into this saying, I don't have enough room to explain this, so I'm only going to explain 1 or 2 of my ideas. Write it all. That for an NIH grant, that might mean that your first draft is 15 pages, 16 pages. It definitely should be longer than those grant requirements. And then after you've written that first long, wordy, unconcise draft, then you're going to be editing that. And you use that editing process to remove redundancy. I find that the vast majority of scientists, including myself, you tend to write our first drafts redundantly, use way too much passive voice. So there's a lot of, it has been shown that, instead of just saying it is. And by going back and removing all that redundant writing, you really, really can clean all that up in editing and really shorten the grant. And this is a place that there's an article of one of the articles that I gave Erin to post for you. It's called Polishing Your Prose Word by Unnecessary Word. And that's an extreme case of somebody who wrote a page-long document that got down to 1 sentence. Because it sounded all important, but if you really analyzed it, it was just a bunch of redundant writing. So, definitely don't try to write to page limits. Write it long and then edit it back short. And that's all I have to say about the research plan. Now, I think somebody had asked a question about preliminary data and a preliminary data section. Did I answer that question? Or are you surprised to hear what I said about it?
Oh, that was me. This is Evan. That was me. I am currently writing a grant that's for the American Cancer Society, not the NIH, and they had a preliminary one.
Gotcha.
My NIH grant is coming later, so I had read one, and at the time I had read some K awards, which is what I'm writing, and I saw it, I didn't appreciate it, and you totally reminded me. So sorry for opening my mouth like an idiot.
No, that's totally fine. And just actually, even one thing to think about in your in your American Heart Grant, as you're developing that preliminary data section, that unless they say otherwise, it often doesn't hurt to sprinkle some preliminary data in the research plan, or even to just say, as I showed in Figure 3, that such and such and such and such is true. And it's just human nature that grants are often very dense, and you want to constantly be reminding that reviewer, don't slapping them over and over again, that it's really that I have the preliminary data for this. Really, 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 really remember this when you're reviewing my experimental plan. I'm not just making this up. I have data for it, because it's one side on the writing side. You can say, I put it in there. What did they mean? But if you've done a lot of review, you realize that it's really, really hard sometimes to remember what's in the beginning of the grant when you're reviewing the last couple pages. So, to put in things in your writing to try to keep reminding the reviewer of these things, it prevents you from being that person who gets back negative reviews really, really frustrated by the idea that I did that, but they didn't read it. No, they read it. They just didn't remember it because there was too much of a gap. Does that kind of make sense as a philosophy?
Yeah, that makes perfect sense. Thanks for explaining.
Okay. So is there any other questions or comments? I'm happy to take any. I actually prefer the asking questions bit than the lecturing part, so I'm completely open to questions. Or if people don't want to ask, if people don't want to ask questions right now, feel free also to email me. I know there were a lot of people on the call that aren't the direct IMBRI, not Brandy, I'm wrong grant, CTR awardees, but I'm always happy to talk to people about stuff. I have various professional development hats in both IMBRI as CTR, so if anytime somebody who just wants to talk to somebody who might have a different point of view than their mentor, I'm always happy to do so. So feel free to send me an email and we can set up a time to talk in person, okay? So with that, I think I'm going to stop sharing my screen. And unless there's any other questions, I'll see